listening to The Switch. The Switch is a podcast about ideas and experiences that change our minds. I'm Chase Harris, joined as always by my co-host, Alex Berner. In this episode, we speak with Dr. Joan Nichols. Joan is Associate Director of Research at University of Texas Medical Branch, where she is also a professor of microbiology and immunology, and she's part of a research team that has recently completed a pilot study bioengineering lungs. In this conversation, we talk about Joan's story and how she began working on this project, as well as the basics of bioengineering and lungs. We also take some time to talk about the most recent study her team completed and what implications it has on the future of organ transplants in humans. Also, there are a couple of moments in there about perseverance, leadership, and the value of teamwork.
If you like what we do here on The Switch, the best way to support us is through Patreon. For as little as $1, you can become part of the community. Ask us questions, suggest topics, and directly communicate with us. You can also unlock bonus content, like extra questions we ask our guests. If you want more of The Switch, both on topic and off, you can become a patron at www.patreon.com/switch_podcast.
You can also support us by rating and reviewing on iTunes, Google Play, or wherever you listen to us. If you want to connect with us on social media, we're on Instagram and Twitter at switch_podcast, and on Facebook, we are The Switch Podcast. So, without further delay, our conversation with Joan Nichols. I would like to introduce our guest this week on The Switch, Joan Nichols. Welcome to the podcast.
Thank you. Thank you very much for inviting me.
Thanks for being here. So before we get into our main topic and a lot of the interesting things that you've been working on, I want to get a little bit of your background. You know, where do you come from? How did you get into the fields that you've studied? And how did you end up working with the team that you work with now? So start us at the beginning.
Wow. So in the very beginning, when I was in high school, I was invited to a Syracuse University program to work in a research laboratory. And that's the time I think I caught the research bug that made me very interested in doing some type of research. At the time, I was doing organic chemistry research, started out washing dishes and eventually moved to actually doing experiments in the laboratory. So that's a good start. I was a physics major as an undergrad with a chemistry and biology minor, as well as a minor in art studio, to give a kind of a good idea of where I come from along the way. So somewhere in that time frame, I realized that I was the only woman in my physics classes, oddly enough, and not that that was a big deal, but I was also starting to think about getting jobs, and if I'm the only one in the field, it might be kind of hard to get a job. So I decided that although I loved physics, it wasn't the way for me to go, and I made a switch to be a chemistry biology major, thinking I could get a job at the end in some area or maybe go to graduate school. I was very interested in cell biology, took a master's in cell biology, actually at State University of New York in Geneseo, which is one of the SUNY schools. And after my master's in biology, or my master's in cell biology, I worked for a period of time in the Rochester area and at the same university doing research for some other faculty before I decided I wanted to go back to school and get my PhD. So I started my PhD in Rochester, University of Rochester in New York, and I took a program in immunology. And then when my mentor decided to move to Galveston, Texas, he asked me to come along, and I took an entire microbiology program at University of Texas Medical Branch in Galveston.
Wow.
So I have way more education than any one person should have.
That's a good thing. That's an asset.
Yeah.
It's an asset because at first you think, wow, I'm never going to touch this ever again. Wow, this wasn't really important to me. Wow, why did I take this route? And so, but over time you need to realize that the fact that I know physics means I can look at shear forces and what cells might, see in a vascular flow as a cell moves through your blood vessels. Because of my chemistry background, I can look at what happens with oxygen as how oxygenation of a body works and how at the cellular level cells pick up oxygen and manage it. In the biology level, I can understand how cells function, how they come together to make tissues, how your tissues come together to make your body, and how an organism works. So, including the art studio work, because I do all my own graphics for what we need to for our publications, it eventually works out that you have all what you need.
Yeah.
You just don't give it as a plan.
Yeah.
Yeah.
Yeah, that's something we've noticed with some other guests as well. They have interdisciplinary skills, and that's really what's led them to be, unique in the perspective that they have on each topic.
And successful in whatever field they end up in.
Mm-hmm.
Yeah.
That's true. And the more things you do that are different, the more experiences you draw upon to make you be what you are and what you're doing. And the art background also helps in a perspective that says, I can appreciate it. It's an artistic standpoint of looking back at the research that we do that says I can bring art and science together in a way that makes people excited about it.
Mm-hmm.
Cool.
So that's what I do.
Awesome. And so that's how you ended up on the team that you're working with now?
That's an interesting story, and that's one of the things I was going to describe as we move to the end. But
yeah,
a team like this doesn't just happen. A team like this, the way we've come together is in 2001, I met my now research partner who is a MD. And I had been doing work to model the human lung in terms of looking at respiratory pathogen responses, which means I could take these little balls of lung cells, I could expose them to a virus that might cause a disease, and I could watch what happens. Mm-hmm. My research partner that I met is the person who came in and said, and he had some background in doing engineered trachea, so bioengineering tracheas for people, knew about scaffolds. And it was kind of like, oh, holy cow, this is the piece that I need to have to understand how to go to a new level. Mm-hmm. And he was also the one that pushed the idea that if you can make a quarter-sized piece of lung tissue, why can't you do something bigger? And it doesn't always work that way, but it's a good place to start from.
Yeah. So could you sort of briefly describe, like, elevator pitch what your team does and your role within the team, and then we're gonna back up and get a little bit of groundwork done before we launch into the most recent developments.
That's great, and it's a good place to start because I do a lot of modeling of the lung, cell biology based modeling, where we construct small pieces of lung tissue to evaluate physiology. What does the lung do? How does the lung do it? What are the products the cells that are in that piece of tissue make? How does that influence something bigger? And in understanding and modeling what the lung does in terms of a small three-dimensional model, you can do a lot of repeated testing. I can look at how a nanoparticle or a hydrogel material, and hydrogels are the stuff that makes your toothpaste thick. They're thick gels. And I can put factors that help cells know what they need to do, know how they need to function, induce them to proliferate, make more of themselves. I can use these factors, they're called growth factors, in nanoparticles or hydrogels. And I did a long series of tests over the years of understanding if I apply a growth factor in a certain spot, what happens to the cell. If I have a nanoparticle that releases the entire payload of its factors at one time, why is that different from a time release of factors? How does that influence tissue formation? So if you understand where I'm coming from, it's all of the small little pieces like building blocks that you need to work with to understand, well, a group of cells hanging out doesn't make a tissue, but a group of cells on a structure, a scaffold material that gives them a dimension to work in, entices them to act more like a tissue and grow together. And with the factors that I deliver to those cells, they know exactly what they should be doing, and they respond in specific ways. And it's like training a puppy. You start with telling the puppy this is what is reasonable for you to do, and then you entice the puppy. You never make it to go where you want and potty train and the whole bit. The same thing. You entice these cells gently, to do the things that you need, attach in the ways that you need them. You can never make them. That's an engineer's perspective, that we do something, we make it do it. No, we entice them to go where we want and function in a way that's beneficial. And for a model, what's beneficial is to act like the lung or a trachea or a bronchiole or a piece of your respiratory tract. Because remember, when we're talking about the lung, it's bigger than just a lung.
Yeah.
So does that give you an idea of what background?
Yeah.
That's what we came from. Looking at wound healing, looking at if you injure your skin, how does your skin heal? What factors do you need to add to heal better? And a lot of these factors are now available in creams for you to work with that stop you from getting scar formation. Same thing we needed to do for our lung study. How can you entice healing in the lung after an injury and not get a scar to form? Because scar formation in the lung is bad. It's fibrosis. And fibrosis stops the stretchiness of your lung and stops you from breathing.
Wow.
Does your team have a mission statement? I want to frame the rest of our conversation around where we're going to end up.
Mission statements. Research teams don't usually have mission statements.
Okay. All right.
Wow. I'm trying to think what our mission statement would be if we had one. How about just something like, although nobody's done it before, doesn't mean we shouldn't be trying to do it now.
Nice. All right.
I think I'm going to write that down, and that's going to be our mission statement. Because that's part of this. Although nobody has done what we've done before, there are a few labs that work in the area. Nobody took our view. Nobody did it from the smallest level. And I think that's what's made us successful.
Yeah. So, okay, let's start at some of the basics. We started to talk about lungs.
Right.
I want to get everybody on the same page with lungs. So can we start sort of at the ground level of what is a lung, what is it for, and sort of how does it work mechanically, chemically, and its role maybe in the broader context of the body?
Wow. Holy cow. So define the organ. Okay.
Yeah.
So if you just sit still for a minute and you breathe, and we all breathe a lot, and we intend to breathe a lot more, but if you just sit still for a minute and you take a breath in, the air that you breathe in goes into your nose or mouth, if you're a mouth breather, goes into your nose or mouth, down what's called your trachea, branches off into the main stem bronchi that feed into each of our individual lungs. They sit right and left in our body, in the upper part of our chest. And as you breathe, the diaphragm, which is above your liver, below your lungs, the diaphragm moves down. Your chest cavity, the muscles tighten between your ribs, and then that expands your chest cavity. And you pull down and actually make that structure that's your chest cavity, pull enough so that you're letting your lungs expand and enhancing that expansion as the air comes in. And so your lungs are basically part of that conduit system to have air come into you. So what is their function? What do they really do? The lung is filled with these little kind of areas called air sacs, alveoli, that are lined with very thin cell types that actually allow oxygen, or as you breathe in, you're breathing in air, and part of that is 21% oxygen. The oxygen that's in the air you breathe into your alveoli passes through those specialized cells in your lungs so that the oxygen moves through the cell into your small blood vessels where your red blood cells sit, so that they can pick up oxygen and carry that with them through your body. Okay? So your lung has this function that allows you to breathe, and this process occurs again and again, that you let the oxygen pass through the cells, move into your red blood cells, and the red blood cells move through your body carrying oxygen wherever you need it. They also eventually, you have a mechanism to bring carbon dioxide, a byproduct that your body makes, that comes back to your lung to be released so that you breathe it out. The process of breathing takes place depending on who you are and where you're located, on top of a mountain, at sea level. We breathe different rates. You breathe differently depending on what you're doing. But this process of breathing is what brings in the oxygen that supplies all the cells in your body with the oxygen they need to survive. And that's a big function, but it's very mechanical.
Yeah.
Based on the ability of your lung to be strong. And so the proteins that make up the structure of the lung itself, not necessarily the cells make these proteins, but what makes your lung strong is a material called collagen. It looks, if you look at it on a specialized microscope, like wavy curly hair does. Okay. And so it also is very strong material. It comes together in bundles. And so we breathe a lot and can breathe a lot because of the collagen. There's also another protein that's in there that's really important called elastin. And elastin is elastic like rubber bands. And it feeds through, kind of like you were knitting a structure. Elastin feeds through that knitted structure that's your lung, the skeleton of your lung anyway. And elastin feeds through it in a way that lets you be elastic. So you don't have to force the air out of your lungs, because the elastin pulls in gently as your diaphragm moves back. And so you actually let your lungs come in and you release the air out of your lungs, and then you breathe again. The elastin lets you expand, and the collagen provides you the strength to breathe through the rest of your life. Mm-hmm. And so basically the function is just gas exchange. There are a lot of other things your lungs do. I don't want to belittle that. Basically, I'm an immunologist, which means I study how you get sick, how your body keeps you from getting sick. And so there's a lot of immune response stuff that your lung does. But at the end of the day, if you really want me to just ignore everything else, just say it lets you bring oxygen in.
So I'm trying to decide whether we go into the idea of organ transplants next, or we go into the idea of bioengineering next. Which do you think would be a better route to follow?
I think what you should do is just general bioengineering, but I want to talk about the stepwise approach to where we got to today. Okay?
Okay, great. So bring us through what is the process of bioengineering?
So bioengineering at its simplest level is taking a cell and a structural support, we call it a scaffold, bringing them together to create a three-dimensional tissue. That's bioengineering. What cell you choose, what scaffold you choose is pretty much up to you. It depends on what you're building, and how big it is depends on what is your end product going to be? So for us, if we're going to build a small structure for modeling, it may be about 1.5 inches in size, or about the size of a quarter. Really, we can make them that size consistently, and that would be for looking at just a model of how lung works.
Sure.
Okay, so that's a simple bioengineered material. So in terms of bioengineering something bigger, though, it's a different story. So if I'm going to take us from what bioengineering is in terms of the simplest term, I need to tell you a little bit of a story about what we've been doing in our research team for bioengineering. So if I go back in time 15 years, I'm growing lung cells in my laboratory. I meet Dr. Joaquin Cortiella, who is a clinician with a background in scaffold material, and we start talking about growing larger pieces of tissue. So we publish a series of papers that talk about different kinds of stem cells, embryonic stem cells, adult stem cells from the human body, primary cells, and primary cells are cells that I might get from a piece of tissue, a piece of lung tissue. Could be an animal, could be human. I'm fortunate that our research team is allowed to get human materials to study. If tissues aren't suitable for transplantation, they're made available to research scientists, and so I can get human lung cells if I want to study them. So we published a lot that said we knew how to isolate these cell populations, we knew how to grow them, we knew how to entice them to do the things I've talked about, how to go from just a group of cells to making a tissue and to making proteins that would be specific to the lung. And then that was very nice. That was really good, by the way, but not far enough to give you an organ, not far enough to take you towards bioengineering an organ that might be useful for clinical application. So the next round of publications came when we realized that a lot of the scaffolding materials that we work with are very boring. They're made out of one product. They're made out of gelatin. They're made out of just collagen. They're made out of other materials like suture material. There's something called PGA, that's polyglycolic acid. Doesn't matter if it's a big long word, but it's suture material that if you had a surgery, they would use to stitch you up. Cells like to attach to it. So we could use these materials, they're very simplistic though, to grow, I would say, a three-dimensional structure of tissue, of lung tissue, but it didn't have any specific shape and it really didn't have any definition. And so we were looking for what would be good to use as a lung scaffold. So at about that time frame, other studies were happening and scientists were looking at better ways to make natural scaffolds. And so we developed a mechanism in our lab to take a whole lung, whether it's for a pig or a human, and remove all of the cells, all of the blood from it, and only leave behind those structural skeleton proteins of the lung, the collagen and elastin. That's a scaffold. It's called a lung scaffold, and it looks like a lung does. If you just Google my name, Joan Nichols at UTMB, you're going to come up with all of this information as we move along. But the fact that we came up with a process to remove the cells and the blood from a lung meant we could take a human lung not suitable for transplantation, take all of the blood clots out of it, and remove everything and just leave the nice gauze-like material of the scaffold behind. That's your structural basis to build a new lung. And so we publish this information. By the way, the process we use is detergent-based. It's a sugar first, like glucose, sugar, what you eat off what you have on your table for your coffee, and detergent, like dish soap. Well, okay, it's not exactly dish soap, but it's the stuff we make dish soap out of. And this mixture, just like it washes your dishes, the detergent destroys the cells that are dead anyway, removes them from that scaffold, and the sugar is just there to help the collagen and elastin proteins survive the detergent process. Because you realize it's not just like washing a plate, we've got proteins here. And so once this is done, we have this whole scaffold, this whole skeleton of a lung that has a trachea, that has the main stem bronchi, that has a lung on each side, that has a branching airway, although no cells, and the branching vascular system, the blood vessels of the lung, oh wait, no cells. So it's not really blood vessels. It's just scaffold material.
Yeah.
And that's our building block to start making the next step. Okay, so now I progressed you through 10 years of research in a couple minutes. And then comes on the last 5 years where we were working hard to actually prove that we could use a whole organ scaffold and put cells back onto it to recreate a lung. We published a paper in 2016 that took a pediatric sized, a small child scaffold. The child was killed in an accident, and the parents asked the people who came to procure, it was too damaged to ever use for transplantation. So what they did instead was the parents asked that something good come of it. And so that was brought to us by the cardiothoracic surgeon that we work with. His name is LaFrancesca. He's an amazing surgeon, but he drove this down to us so that we could have that be the first lung that we took all the cells out of in terms of a whole organ, and that we took cells from an adult lung that we had already stored in the freezer. We brought them out, and that was the first lung we reconstructed. So that's what we published, production of a pediatric lung. So we put the cells where they needed to be. We put the vascular cells, the cells of the blood vessels where they needed to be. You can argue, as did many scientists that are in the field, that's not a bioengineered lung. It doesn't prove anything. It doesn't. It was really beautiful, though. It had the same work of breathing that a normal lung would do. We developed a method to pump through a material that carried a dye through the blood vessels to prove that the vessels didn't leak. We did as much as we could with that, to the point that said this is not going to be transplanted anywhere. And we did that with 5 other lungs as well. But we weren't ready because they hadn't been tested well enough to prove they would work on a person.
Right.
Hence the reason to do a pilot preclinical study. Preclinical means before it ever goes to the clinic, in an animal model, which is required by the Food and Drug Administration, so that we prove that, and I would like to do this too, I would like to prove that this works before I ever approach a person and say, hey, you need a lung, I got a lung I can make for you. Okay.
Yeah.
To be realistic, you want to step in between. Okay, you're not ever going to say to me that you guys are willing for me to transplant a lung into you at this stage, right?
Right.
Good. You think well. So that's coming to the present day study that we did, which we actually started years ago with the first animals that we did, where we developed lungs for pigs. And so back up in time, remember we've already published how to make scaffolds for pigs or humans, whole lungs. We did a pig study that would prove feasibility of doing this. So the pig study went like this. We knew the lungs that we were creating weren't mature enough, hadn't developed well enough in 30 days that we culture them in a bioreactor. Bioreactor is just a big tank that we run lines into to pump fluid through to keep it bathed in a fluid that provides nutrients to the developing tissues and keeps it alive. And oxygen too, we pump oxygen through to it as well. So we have an immature lung that we know that we want to work with, and so we developed a process. Because of the questions we wanted to answer, we developed a process that went like this. For each animal that we worked with in the study, we took out one lung. We took out the left lung. And we used that lung as a source of cells. And on an unrelated scaffold from another pig donor, from totally unrelated, we could even buy lungs from a butcher shop and produce the scaffolds that we need. We took the cells from that pig, an unrelated scaffold, put the scaffold in the bioreactor, put attachments to it, and I don't know if you've seen any of the pictures that shows tubing running into the trachea and the pulmonary artery and the pulmonary vein, into the vascular system and out of the vascular system of the lung and into the lung itself. And we pumped in cells. First, we started with vascular cells because in 15 years we learned you can't do the lung first. We're not building a bioengineered lung. We're building a bioengineered lung vascular system that we need to put lung tissue around. Mm-hmm. So in the 30 days, we start with the vascular cells first. Growth factors in hydrogels and nanoparticles go in on a regular basis at the beginning. The cells go in. Every single day, there's a recipe that I follow that I look at and says, this day, this kind of cell, this speed to pump it in, how long the pump is shut off once we pump the cells in so they can attach. And then you go to the next one every day until at the end of 30 days, we made a lung for that pig. Belonged to that pig was that pig's cells, and that lung went back into the same pig that was the donor. And then we survived our pigs for different periods of time. And at the end of the time frame, because we didn't know what the lungs were doing, we had to euthanize each animal at its time point and look to see what was the tissue doing, how well did it develop, was it developing vascular tissue, was it connecting in well with the vascular system of the animal because we disconnect this and we're letting it connect naturally, was it surviving, was it creating any problem for the animal, because we didn't want to make the animal sick. And even though we could image, we can do CT scans and x-rays, you really can't tell what's going on unless you look at the tissues. And so at 10 hours, 2 weeks, 1 month, and 2 months, we euthanized the animals to look at what was happening with the engineered tissue. And so that's what this pilot study was about. Now, the questions that this pilot was supposed to answer went like this. The first big question is, if I took cells out of you, grew them for a month in a what amounts to a big jar in a laboratory, kept them alive, put them back into you, would your body say, welcome home, I missed you? Or would your body say, wait a minute, you don't look the same, I don't recognize you anymore, I'm going to reject you? So that was the first big question. The second one was, would the microvasculature, so the little tiniest level of the blood vessels called capillaries, would they form properly so they might be able to support oxygenation, which is when you breathe and the oxygen goes into your red blood cells. And the third thing was, are we going to cause any harm to these animals that we're working with? Are they going to be fine? And so I'm glad to say the first question, there was no rejection. The second question, everything went well. The tissues continued to develop. And in terms of what we saw, we had good microvascular development, no leaks of any of the blood vessels we created. The animal's blood supply connected in with the blood supply of the bioengineered lung. And by 2 months out from transplantation, the animal's tissues started to really look like mature lung tissue. We also learned a lot of things that we didn't really know before. That some of the things we didn't really have good questions, that when we came back later, we're like, wow, we're learning all this other stuff. How to do this better? What do we really need from the equipment that we have? Because the one thing I neglected to tell you was, by the way, you can't open a catalog and buy this equipment. Mm. We have what we call Fabrication Friday kind of events where we make what we need. So the first bioreactor we ever made to take the blood and cells out to produce scaffolds was a fish tank from a pet store, was plumbing from a hardware store, your choice of, because I think we've been to Lowe's and Home Depot and most of the major hardware stores. Boat plastic, because you can saw it any way you want to and sand it really easily. And the pumping system for the fluidics, the lines that ran into the tubing that runs in between the system, was bought on eBay. Wow. So nice. Yeah. So yeah, make what you need as you need it. By the way, if you go to Home Depot and you're going to do a project like this at home in your garage, don't tell them what you're doing. They tend to not want to wait on you anymore. So just say, oh, I just need some plumbing fittings, and just go and dig around yourself. So, so that's part of this 15 years that led up to this project prior to this, because you have to make your equipment, you have to make the recipe. And just like baking a cake in this study, we wanted to prove that if we followed this recipe, we could produce a lung that was similar. Just like buying a cake in a store, they all look the same. We wanted to make sure they were as close to being the same as a product as it was possible.
Mm-hmm. Yeah.
So that's where we are today.
So you mentioned that you learned a lot of things. What was maybe some highlight that you learned that was counterintuitive to you? Something that went against your intuition for what you were going to see?
Wow. That we really knew what we were doing with these nanoparticles, that they would release the growth factors in a time-release fashion in the blood vessels, and that we would get the microvascular development. Modeling it looked good, but making something in reality that connects well enough with the body, a whole different story. And so what we transplanted into the animal is a scaffold with, that's immature, but it has so many growth factors that we put into it that those factors really help to drive the development of the vascular system from the pig to the lung in a way that was amazingly normal.
Yeah.
That's what it was, a surprise to us. What the pressures were that a bioengineered lung was going to have to withstand eventually, because your low pressures in the capillaries where gas exchange happens is okay. That's easy to do for cell culture. High pressures, like what you have in a pulmonary artery, we knew we weren't capable of having that, so we didn't connect that to start with. But we've learned now what those pressures are, is that heart beats, how much you're pushing. When I talked about physics and stress and shear forces and things, we know what those pressures are, and we know now how to develop a system that will mature those vessels to meet those pressures, starting at really low, just gentle streaming of cells into this, to really high forceful pressure, pushing a lot of cells into this organ because it has to oxygenate your whole body.
Mm-hmm. Yeah.
Those were cool things we learned. We also learned something that part of the project, and not many people have commented on it because this is a really science heavy paper that I'm describing to you. But we also looked at the microbiome. Your lung has a microbiome, organisms that live in it, microorganisms that are there all the time. In this project, we actually wanted to know how quickly your microbiome reestablished in the engineered lung. Realize the engineered lung is sterile in its tank until the first time that animal breathed. And then it's dirty. So we wanted to know, would you get a bad microbiome building in that lung, or would it be normal like the rest of the animal's respiratory tract? And because we knew it was going to have its own microbiome, we gave that lung back a very rudimentary, very basic immune system. We took white blood cells that are the cells that are part of your immune system that keep you from getting sick. We isolated them from the animal, and we gave them back to that lung just before transplantation. So there were immune cells waiting, poised. When that first breath happened, the immune cells were where we needed them to be, that if any microorganisms had broken through into the tissue itself, there would have been an immune response that kept any infection from happening. Because unfortunately, with lung transplants, many of the transplants fail because you develop an infection. That's why understanding what the microbiome was it normal? And did the immune system help protect those lungs when the animal breathed? So now you know why it was a pilot study.
Yeah.
Right.
So how does this fit into the broader context of our sort of global knowledge of organ transplant in general? Are there other people sort of trying this scaffolding plus growth factors approach?
A few people. Not the way we did. I want to talk about a difference. Like I said, I think everybody, including us, and we failed a lot of times growing a lung by focusing on the lung. When we switched to focusing on the blood vessels, though, was when we made a switch that was critical to us that changed our whole process. And it may seem like something small to you, but if I'm telling you you're going to engineer a lung, you think, oh, I want the cells that are involved in gas exchange. That's what a lung does.
Right.
No, you want the blood vessels to be perfect so that you can support gas exchange. And so that was for us a big switch that we made, and my students hate it. By the way, students hate it when you say to them, we failed. They will not say that word when they're talking about their research at that stage. I say every day, well, we failed. Throw it away. This fluidic system, the tubes that we've got connected to this, clog every 10 minutes. This isn't good. Change it. So we're constantly modifying till we came to the point where we said, I think we've got the best thing now, let's go ahead. That's different from other people in the field, I think. And our approach, too, of looking at it from a view that says the physics is important, the biochemistry is important, the cell responses are important, the scaffold is critical. And this is the step that we're making today to come to the point to do this process. But it's not the end process. It's where we are today. So if you look at that timeline and you say, in the 60s, we started doing organ transplants. Okay, in 1954, we did the first kidney transplant in a person. And then in the 60s, we started doing liver and heart transplants. And then in the 80s, we started to do lung and intestinal transplants. Before that, the big deal was if your organ wore out, you died. Bringing in basic transplants in terms of donated organs was a good step. Our next step is taking all of those. By the way, we have to have a scaffold from an individual and cells from an individual to bioengineer a lung for a person or a pig. But for people, half the donated organs that are collected can't be used. They're discarded or they're given to research because they're not good enough to transplant. So if you've got people waiting for 3 months, 6 months, years on a transplant list for an organ, we're throwing a lot of organs away that we say, okay, we know how to do transplants. We know we can grow an organ that looks like the organ that you need. So a surgeon doesn't have to learn a whole new process. We just need to have these discarded tissues to work with to do it. that's a nice next big step into supplying organs eventually for people that need them.
Do you need a human scaffold to create a human lung? Or can you use...
We did make a human lung with a human scaffold. We may be able to make a human lung using a pig scaffold, but that's not something we tested here because we made sure the scaffold and the cells matched for the pig study. But it's nice. That's something about in the future might be looking at it that way and saying, can I maybe doing the opposite? Take a human scaffold, put pig cells on it and put it in a pig? And say that's about the same way as if I do it the other way around for a person. But I think we have enough scaffolds that would be available right now because, again, this is just a turning point. The next big step is going to be I take a small biopsy from you. I either make you specific stem cells that are you, or I've gotten to the point where now I can actually expand your cells, make more and more of the cells that I took from a biopsy, and I bioprint them. After doing a CT scan of your chest, I bioprint a lung that looks just like your lung, matches your lung with your cells, no immune rejection, and we give you a new organ. So in terms of you talk about switches, well, the first big switch was doing organ transplants, having live people come out of it. The second big switch is what we're talking about now, producing bioengineering for people who need these tissues. The next big switch, and I hope to see it in my lifetime, but probably it'll be my students, is bioprinting an organ as you need it to match you, no immune suppression at all. Don't have to tissue match you because we know my studies prove we can grow cells of yours outside of your body for a month and give them back to you and your body welcomes them back. Okay, bioprint it and the lung is yours and give it to you when you need it. You don't wait on a transplant list. You just wait for the time that it would take to grow it. And it may grow longer than a month, but it's going to grow and it's going to be yours. And it's a heck of a lot faster than waiting for someone else to die to procure an organ that hopefully they signed an organ donor card that says that we can procure their tissues and use them to save somebody else.
And hopefully it's suitable for transplant.
Hopefully it's suitable. That tissue match thing that we've avoided, because that starts to get a little sciencey there, that says you have a tissue match. It is specific to you. You get half of it from your mother and half of it from your father genetically, which makes you very special. By the way, be nice to your siblings because they're closest match that you've got. If you don't have siblings, be nice to your parents. They're the next closest match that you've got. And if you have an identical twin, be real nice to that identical twin. They're a source of anything you need along the way as you live your life. Okay?
Mm-hmm.
But think about the fact that tissue matching is hard to do. So what I'm talking about right now until we can bioprint is taking cells from lungs that can't be used, that have live cells in them, taking those cells out and banking them. And I know what the tissue match is going to be. And I bank those cells. And then I have this huge facility that's full of freezers, full of bank cells, and you need a lung. And I say, okay, you don't have a brother or sister, do you? So you're kind of stuck and you only have your parents and they're not the best match. Let me look at my bank and I can come up with something that might match you as close as possible. And I would tissue match you and then take those cells and either bioprint or take a scaffold from another person right now. Put the cells on the scaffold, grow you a new lung, and there you go. You're at least closely matched and maybe don't have to be as immune suppressed as if it was just an unrelated organ.
I'm wondering specifically, what is the next step to getting us to where you just described?
The next step is a long-term survival study where I do the same experiment that we just did and produce maybe 8 lungs for 8 pigs, specific to them, do the transplants for those pigs, send them back to the farm for 6 months to a year to have a nice little piggy away, the life on the farm. And then at the end of that time, bring them back, anesthetize them, block off their normal functioning lung, and let them just breathe on the bioengineered lung.
Yeah.
Prove that we have gas exchange and enough Because you can live off one lung, so can a pig.
Mm-hmm.
Prove that that animal can live just with the oxygen supplied from the bioengineered organ that we created.
Yeah.
And as soon as we can get funding, that's exactly what we intend to do.
Mm-hmm.
By the way, the earliest I could submit for the next round of funding would be October, which would mean April if we were accepted and granted funding from NIH or NSF. If we were granted this, remember there's a big if there, we wouldn't start until April of next year, but that would mean sometime in the next 2 years we could answer that last piece.
Mm-hmm.
And if it didn't mature enough, we would at least know what we needed to change to make this better.
Yeah.
Right.
Yeah,
And
absolutely.
by the way, as an aside, what if someday you really want to go out there in terms of science fiction, what's next in medicine?
Yeah.
What if I learn enough from doing all of this bioengineering that I come up with a nanoparticle treatment that helps you grow your own cells with your own scaffold in your body? And we entice your own body to recreate what it needs to repair itself.
Yeah.
Wow.
We're talking about wound healing on a scale, not just where you heal a cut. We're talking about wound healing regeneration of tissues, like growing a new limb,
Yeah.
grow a new piece of an organ that you need.
Wow.
And that's also possible because the same studies that we're doing now to bioengineer, we're learning what you need to do to grow the tissues and do everything that I'm talking about. It could be eventually something we apply just to you and how you function.
You've been very good at anticipating all of the questions that I had written down. There was basically a big list right in a row where I asked you the first one and you went straight through, which was great.
Probably because they're the questions I've been asking myself.
Yeah.
Mm-hmm.
What led to this? How did this come together? How did the pieces that were, that for each of the people, in terms of this whole research team coming together, we met the right people at the right time.
Yeah.
I'm not saying a higher force influenced us, but wow, something really set this up so that I met my research partner at the time. I maybe could have figured out how to do some of this, but no, not without that input of somebody with a piece of information that I wasn't even trained to look at.
Mm-hmm.
Our cardiothoracic surgeon I met at a meeting. He chased me down. I presented at a meeting and he chased me afterwards. And he came up to me and he says, "I'm a surgeon. I love what you just talked about. I would like to be involved in your project." The people that make the nanoparticles that I work with at Methodist Research Institute, Jason Sakamoto, I met him because I applied before anybody really knew what nanoparticles were. I applied for a grant to enhance wound healing way like 10 years ago and met Jason Sakamoto at a meeting, but because he was running the meeting and was working for a center that was trying to enhance people's understanding of what nanoparticles were and how to work with them.
Mm-hmm.
And so he's involved in making our nanoparticles and helping with that side of it. We met the right people at the right time, including veterinarians that were willing to look at this. You realize there were a lot of people that said to us, "You're going to kill all of the pigs that you work with. This is not possible." We've had people in meetings say, "I don't believe your research data." That's normal. When you push an envelope, people don't like people who push an envelope. They like you to make little steps. Big steps aren't received so well. So I'm lucky that we met a group of people that looked not at the things that would fail, although we knew we would fail, but also said that the only way to learn what to do is to fail.
Yeah.
And when you fail, you get up and you figure out what you need to take a next step. So just like making the light bulb, and Edison and all that process, many, many ways how to fail. One way that eventually worked that you succeeded, and now lots of ways that could be potentially successful.
Yeah.
That's how a project like this works. So meeting the people when you needed them and have them be as crazy as we were, as willing to push the envelope. It takes, I'm not saying crazy, but I'm saying, right. I don't have a good term for it, but a kindred spirit who's willing to say, we're all going to hang hands. We're holding hands at the edge of this cliff. Close your eyes. Ready? 1, 2, 3. We're jumping off together. Don't look at what's at the bottom. Mm-hmm. And a group that's willing to do that and trust you is something that's amazing to behold. By the way, this project was done with very little funding, ridiculously little funding. It paid for the pigs and the growth factors, nobody's salary. I couldn't afford in a grant probably to pay for the salary of all these clinicians anyway. Our anesthesiologist, Adam Wacher, is an actual clinical anesthesiologist who provides anesthesia to patients receiving transplants. The surgeon was, I mean, he's the guy that was actually putting the lung in place. He flew himself when he was in Houston at first, and then he moved to Boston. He flew himself from Boston on his own dime to do our surgery. And my research partner, Joaquin Cortiella, who was scrub nurse for most of these processes because he gave up anesthesia to somebody who did transplant anesthesia, but he's a pediatric anesthesiologist, but he ran the show in terms of what the transplant team was doing. And managing and getting these people together. People who donated their time and all took a vacation day, so they were compliant, all took a vacation day to do the transplants. I mean, that's an amazing research team. And on the research side, students, graduate students, and people who are willing to get up in the middle of the night or sit and watch our tank that had the lung sitting in it, culturing for 30 days. We didn't have a camera assembly on it. We didn't have enough money to buy one. We could only watch it. And that meant physically getting up in the middle of the night if I was worried about it, driving into the laboratory, going into the lab, opening the door to see that the tank was leaking again. Or something, the pumps had stopped working, or something that would mean we wouldn't have a lung. And by the way, every animal that needed it, we always produced a lung for it. So an amazing group of people that pulled together to do something almost impossible. No weekends off, no holidays for a couple years at least, all pulling together to get this done for just this small pilot.
Yeah. So how many people, do you know how many people need lung transplants, or who, basically how many people could be affected by this, once we get to being able to do it for humans?
I've been trying to get a number of how many people need a lung. What I can tell you is there's a lot of data about the fact that there are increases with people with severe lung disease worldwide. We know there are not enough organs to go around, and that's why people have to wait. The actual number is harder to get at, though, because we're talking about cancer, emphysema, COPD, fibrosis, pulmonary fibrosis, many conditions that end up for somebody that, and the serious stages, I mean, there's no option but a lung transplant. So, but numbers, I've been looking, numbers are kind of hard to get on it. And I'm sure somebody that listens to this is going to end up letting you guys know there's a number.
Right.
But I didn't come up with them.
Yeah, okay. If someone does, we'll put it in the show notes. Along with many of the studies and videos that you've mentioned so far, we'll have links to all of that in the show notes.
I'll send you a list of what we have. Definitely this publication and a reference list of other things that would influence, that people might want to look at and say, hey, and why do things take so long to be applied clinically? 15 years worth of research on one project, step at a time, many failures, getting funding, maintaining funding. We're been funded a lot to model the lung, not to bioengineer it until a small pilot grant was provided to us to buy the pigs, but a long period of time just getting to the point you're ready to jump off and do this.
Yeah.
So first use. So let's talk about if we do our preclinical testing and we prove this is possible, when would it be applied to people? Well, when and how is the interesting concept. Probably the first use is going back to those pediatric lungs we made. There are conditions where the lungs don't develop in the fetus. One is called, in one, the diaphragm doesn't develop. And so because the diaphragm doesn't develop in utero, the intestines move up into the chest cavity and compress the lungs from developing. It's called diaphragmatic hernia, but just say, the diaphragm doesn't develop. In severe conditions, these babies don't survive past birth. So we're talking about making a small pair of lungs for either a in utero transplant, maybe a biopsy from the mom's cells, or we've also done studies to look at what happens with cells you might collect from amniotic fluid and other people have too, maybe entice them to become lung cells. But bottom line, using our current techniques, we could make a lung using adult cells from the mom and grow a set of lungs for that baby that our surgeon says even in utero is possible. When we transplant in utero, they're doing heart surgeries this way. Give that baby a set of lungs, repair the diaphragm not closing, and let that baby develop in utero and be born with a set of lungs that grows with that baby. That's called compassionate use. No other possibility of you surviving unless you use what we provided. That would be a first use. When? Based on funding, between 5 and 10 years from now. But I think that it's going to take that long because we need to prove that without a doubt, we can consistently do this again and again and again without a glitch.
Yeah.
You know, you can't hide anything. Like a cake that's bad, you hide it with icing. We can't hide anything. It has to be perfect, has to function, has to meet that purpose. And, you know, somebody's life's on the line, so it better be perfect. And that's why this process takes a long time to get to the clinic.
It seems to me that, you know, this type of research looks so promising and looks so just useful in the grand scheme of things. I'm wondering if you could sort of talk more about the hurdle of getting funding. Where does funding come from? Why would it be difficult for a project like this to get funding?
So in terms of a hurdle for funding, the major federal sources are going to be the Department of Defense, what's called DARPA, which is Advanced Research Projects, again, Department of Defense, National Institute of Health, National Science Foundation. Those are federal funding agencies. Most scientists like me go towards NIH, National Institute of Health. And here's how a study section works. You have a bunch of scientists that are funded that are given maybe 20, 30 grants each to read, which means in reality, they're going to pick their top 2 that they think are worth something. The best ones. And that's not saying the bottom one is a bad project. It's just saying that each individual is going to pick their top couple. And you fight to get those heard and scored in a hearing where we all sit together and we all listen to each other and we argue about it. And of that, only the top ranked few are ever going to get funded. So you may be worthy. You may have good research publications. Your project may be groundbreaking, but there are also 10 other people, 20 other people that's projects are just as worthy. They're not all going to get funding. The money doesn't go that far. That's life. That is any of these funding mechanisms, except for the Department of Defense, who has a little bit of leeway, more about what they may want to fund and how they do it. But you're really competing with everybody else who has, I read grants that are beautiful, well written. The science is wonderful. I'd like to see everyone funded, but we just don't have the federal dollars to do it. And I pay some of those federal dollars too, and I get why we don't have a ton of money to do it. So now that gives you just a taste of what research funding might be like for someone like me. So we write grants. I have funding. We've had good funding in our laboratory my entire life, but I write smaller grants. When we've tried to go in for funding for this project before we proved feasibility, I have to agree with people who turned to it and said, I don't believe they can do it. You gotta prove it.
Yeah.
Because, you know, and if you were if you were someone in a funding agency and I said, hey, I can bioengineer lungs in my lab, you know, in 30 days we can grow you a lung and it works. You're gonna say, sure, honey. You're gonna say, I don't believe you. What's your proof? And the proof is this preclinical study. Small number of animals because we had to keep the amount of, you know, we had a very tight budget, small number of animals, but we needed to prove feasibility. I think this proves feasibility. But there are other projects too that are looking at different ways to solve the same problem that may be at the same feasibility level that we're at that are good. And how do you decide who's going to get funded? You fight it out at the end and what's the best one overall. Who's been the best at proving their point that they can do it, who's published enough to do it, Who's proven they have a track record managing grant monies that says they're worth investing in? And that's how funding works. And where I'm not always happy, but it's the life you live.
Mm-hmm.
Do you think that there is or should be a role for public opinion when it comes to which research projects sort of, get the thumbs up from funding? Like, should people who are not part of, say, the medical research community be paying attention to what projects there are and bringing them to public opinion? Or is it really something that should be internal to the field, and just the publications are just there to sort of excite public opinion for research on the whole?
That's a really good question. I believe that people have a right to influence what we're doing. But here's the caveat. An entertainer who has a terrible disease can influence this in a way that only benefits that disease.
Yeah.
Or, a lot of things influence funding. You have to admit, if we have an Ebola outbreak in Africa and people are dying and we don't have any treatment for that at all, that an Ebola treatment and an Ebola vaccine is of critical importance to our public health worldwide, that takes a number one priority. An infectious disease that can spread and has, what we see when we see an outbreak in Africa, really important to be able to deal with it. Funding should go into helping with that. Major public health issues should also influence it. Prioritize what you do. You should also have a say. I should have a say in how these dollars get spent, but not in a way that says we get preferential treatment. But, I think as each of us raises, here's Alzheimer's research is really important. This kind of research is really important. New treatments and diagnostics for emerging infectious diseases are really important. How do you pick who's the most important? And I think that falls back to being a scientific review, the way we do it now. So you can influence what what the public says is critical for us to be thinking about, but it still has to come back to saying, in reality, I understand that you're going to cut back funding in an area of research I work in because Ebola outbreak is devastating people. Malaria outbreak is horrible at the moment. We look at this in a reasonable fashion, and that's got to be managed in a slightly different way.
Mm-hmm.
I'm my view, and it may be a odd and kind of limited view, but my view, take into account what everybody wants, but also do it in a way that makes sense. And again, look at public health. What's the most important thing for us to be working on for tomorrow? Because we're talking about biology and therapeutics for medicine. What's important for tomorrow is development of better sources of energy, development of better methods to make your car run better, cheaper, maybe, on a battery, and make better batteries. And so you have to look in a maybe more global view even, unfortunately. Let's back up one more and say, what's important for the world right now?
Right.
But if you've got lung disease, you're going to think, and rightly so, because it affects you personally, you're going to always pick the things that affect you personally.
Yeah, definitely. Well, Alex, are there any other questions that you have on this topic?
Yeah, just what keeps you motivated and keeps your team motivated, and what do you think about when you failed for the 20th time, and also how do you know when to switch course? Like if you were going for 30 years, no progress, there's always probably something else you can try, but when do you decide this approach isn't working, we should drop it, basically?
Ooh, that's a really good question. How long do you hit your head against the wall before you move around the wall?
Right.
Keeping us motivated is the fact that we have made incremental steps.
Mm-hmm.
Keeping us motivated is the fact that a good group of people that are always willing to say, okay, there's going to be a tomorrow. How do we look at this? For me, when we hit failure after failure with developing the lung tissue because we were doing it the wrong way, it was to just take it and stop. We all stopped And we said, okay, we need a break from this. Obviously, let's all take a breather. And while we're doing this breather, let's try to think about creativity, because there's creativity in research too. There's having a good process, there's understanding how equipment works, there's understanding the biology, chemistry, physics of your project. And then there's something that says creatively, how do you look for a window on that wall to break you through? You're not going to be able to get a door in there, but what's at least a small pinhole that starts to let you see beyond the wall that you've hit? And for us, that's actually just stopping and considering everybody's viewpoints. So high school students work in the lab, medical students, graduate students, faculty. We have a lot of people come and go that come in and out and learn different projects. It's also the fact that refreshingly, since nobody's done this before, everybody's thoughts are worthy. And so stopping for a minute and looking at everybody's thoughts about how to fix something, because sometimes you can come up with answers as an individual, and sometimes that gestalt from having a group of people that pool their thought process helps drive creativity.
Mm-hmm.
And that's what I think we did every time we failed. We said, okay, we failed every year. What small piece is salvageable? Obviously, we want to keep using lung cells. Obviously, these scaffolds weren't working. What's in the literature suggests other things. What do we really want to do? Let's be reasonable about this. What are our options? And, we do a lot of, I would love to have a room that is entirely painted with that white paint that you can write on. I visited a company once in Oklahoma that has a room like that. A room that you can go in with markers and you can start writing up one wall and across the ceiling and across the floor and just keep filling it with possibilities and erasing things and coming back until you walk in the room and there's the answer. And I think that's a good example of what we do in our heads when we look at these problems. And I'm not letting you into my head because that would be really scary. But I'm telling you that in my head are these big white walls and areas that actually they're not even walls, they're glass and you can see through, that I write on one side and write on the other side. And I'm always adding the next piece about what I think, we should do. And I'm looking at areas I should erase and rewrite. And so that's how your brain functions when you look at these problems and how you get around them. Does that make sense?
Yeah, definitely.
Absolutely. Joan, are there any other issues or topics in this realm that you think we haven't touched that would be important to hit before we move on?
I think the big thing to realize is that everybody gets faced at some time with things that they think are insurmountable. Everybody has somebody look them in the eye and say to them, that was stupid what you did. You failed. I don't believe you can ever do that. That sometimes those aren't bad things. Sometimes those are things that turn you around in a way that says, okay, I may have failed that way, but I can fix it. I may not be the brightest person in the world, but I have a really good imagination. I can use it. And for every person that says, I don't believe you, and they look down on you because they are so much brighter, smarter, better than you, nobody's brighter, smarter, better. They're just different. And you have to become bright and smart and better at what you're doing to go beyond what a limited view of somebody else has of you. I think that's really important overall, no matter what you do, whether it's research, whether you're artistic and you're creating something, whether you're writing a song to play on your guitar, whatever you're doing, you're doing it to the best of your ability with what you've got to do. And you're the only one at the end of the day that you have to please. Not the rest of the world, not your parents, not your partner, just you. And if you come to that point and you're happy with what you're accomplishing, then you're on your way to being the best person that you can be and doing the most you can. That's something we don't think about usually from a scientist.
Yeah. It's beautiful, though. That's a great perspective.
Wish I'd written it down.
Oh, it's recorded.
It's recorded.
It's recorded. I'll have to play it back and write it down and say, that's really what I think we should do.
Yeah. Keeping in tradition, we almost always bring up some type of Buddhist principle on each show that seems relevant. And there's a attitude of being thankful for criticism. You know, when somebody comes up and says, you're an idiot for doing it that way, you know, instead of having, you know, the typical reaction of, you know,
I know what that
yeah.
Mm-hmm.
Yeah.
I've been there. You train yourself if you deal with people never to get angry.
Mm-hmm.
Unfortunately, once or twice in my life, in a setting where I shouldn't have gotten angry at someone challenging me, I actually did. And you don't ever want to do that.
Right. But if you know, exactly. And in Buddhism, it's when that person comes up to you, you cultivate a mindset of thank you for telling me that. You know, I'll take that perspective into account, and you know, you might think about it and say, I don't think that perspective has much to offer me, or that person was right. But in the end, they're just giving you a different perspective than you have, and hopefully it's valuable in some way.
Yeah.
And in some ways, a drive. I think when people tell me I can't, maybe I'm very contrary. Tell me I can't. Tell me I'm not good enough. Tell me you don't believe what I've done is real, which is horrible for a research scientist. That's questioning your integrity. Tell me I have no integrity. Ouch. But you're right. When people have done that to me, except for those times, and I learned from them not to, I look at it in a way that says, okay, your view, everyone's allowed their own view. But I've also used that to say, okay, you don't believe me? I'm going to prove it.
Mm-hmm.
I'm going to prove it in the best way that I can. I'm going to spend 3 years of my life without any time to do anything else. No holidays, no vacation, no fun. Okay, fun in the lab, but no like vacations or anything. Work Saturday and Sunday. I'm going to do this consistently because I'm going to prove it to you. Turning it around and making it be positive instead of negative.
Mm-hmm.
And you're right. That's an excellent view to have, you know, regardless of your religion. I think that we can learn a lot from that type of viewpoint.
Mm-hmm.
Yeah.
So do you get a break now or back at it?
Actually, 2 weeks ago was the first weekend that I had off. I had Saturday and Sunday off.
Wow.
Cleaned house. I did stuff. Normally my most exciting trip would be grocery shopping over a weekend, which I would get up and do as soon as the stores open at 7 in the morning.
Mm-hmm.
I could wait till later in the day. I don't see people usually when I'm out in the world, and it's a whole different existence. So a little break now, just a little breather now. I don't want to get too used to it because we have this next whole project where we're going to have to prove that we can survive just on our engineered tissues alone.
Yeah. Awesome. Well, I think that's a great way to close out that topic. And with that, we'll go ahead and transition to a segment we like to call the off topic. Joan, I know Alex briefed you on this, but quickly, the off topic is really anything you want to talk about that's not related to our main topic. It could be a philosophical idea. It could be a hobby. It could be a charity. It could be pretty much anything, and we'll spend a few minutes on it. So, yeah, Joan, what do you have for us?
So I have an interesting, and not related to the research, but another thing, the serendipity of who you meet in your life, when you meet them in your life, how long they stay in your life and influencing you, and where that all goes to, I think is a huge, amazing, cosmic question that is phenomenal. So for me, also include for things like that, because I can look at it personally and say, why would I have been a physics major? Why would I have met people that were more biology based? Why was my interest in chemistry so important? Why did I make the transitions that I did, take coursework that I did? I took a year's worth of embryology when nobody took embryology. I had to have special permission to take a year's worth of embryology in my graduate program. I didn't know why I was doing it. I liked it. I grew tadpoles, let them grow their legs, turn into little frogs, took them out and freed them in the world again, because I needed to see how, I guess, looking back on it now, how tissues formed and developed in terms of fetal development. I did drawings and studies of fetal development, watching and reading everything that was available about fetal development. But that wasn't my area. I was a cell biologist at that time. moving on to, as I met people and progressed, as I made changes in what I was doing and met people which seemed to me to be just the right time, meeting my research partner because he came back to Texas and I was dragged to Texas. Okay, I was dragged to Texas by my mentor to finish my program, but I wasn't moving here willingly. We both came from New York to Texas so that we were in the right place at the right time to meet each other. Even bit more bizarre, he moved into the same apartment complex I was in, or he was living there when I moved here. He was in the same apartment complex, but we didn't meet then. We met later when he left Texas to go to Boston, and then he came back years later to Texas. We met because a friend of mine said, hey, I've got to interview this guy at lunch who does research. I don't know anything about research. Come to lunch with us and you can chat with him and tell me if he knows anything. He was being interviewed for a position here. And I said, sure, to my friend, and went and met somebody. And we actually made a drawing at that point of on a napkin of what this project ended up being.
Wow.
2001.
Yeah.
Wow.
Okay. So that, and we still have that napkin, by the way. It's folded up in my office sitting behind me. But
Nice.
we drew out. I mean, it was what I was doing then, what he knew, and it came together. And I have pictures of a little scaffold with cells attaching to it, and our preliminary studies in mice and where they would go to later, and going to a large animal concept and doing whole organ before we even knew that any of this was even remotely possible.
Yeah.
Meeting people as we progressed that provided pieces of information, nanoparticles, which isn't really my field, but getting somebody who's so excited about nanoparticles that, I mean, he lives and breathes it, that influenced us in a way that said, hey, these are going to have a benefit in terms of medicine eventually. How can they benefit us? But coming together with all these bits and pieces to come to a point to do a project like we've just accomplished. That's an amazing kind of cosmic serendipitous thing that said, did this all just happen because it was coincidental? Or did this all happen because somewhere in the universe or the universe itself said, I think it's time that this thing gets done and these people are getting to be in the right place in the right time. It's going to be them. They're going to do this small piece of this big puzzle that is understanding how we function and why we function.
Yeah. My first instinct, especially with sort of smaller cases of serendipity, is to say that I think serendipity is a skill of attention and maybe subconscious effort. The first thing that came to mind when you mentioned it was this concept that Alex and I both learned in school when we talk about video game music. So video games are different from other media because the person who's consuming it actually controls everything. They control the pace, they control, say where the character is moving. And so to write music specifically to what's going on in the game, you have to either limit what the person can do or limit how much the music is actually responding to things in the game. And it's this delicate balance of trying to get the music to sync up with things. And every once in a while you get this thing called serendipitous sync. And it happens in a lot of the greatest games where you're playing and the music just happens to match up with what you're doing. And you can ask the developers, hey, how did you do this? And they have no idea. They're like, we just turn the music on and it works there.
That's funny. And I don't play a lot of video games because I haven't had time to do anything like that. Yeah. I know what you're talking about with that syncing of music with what you've got, because what few times I have done them, there are a couple of them that are, okay, very rudimentary. I'm sure you guys are way beyond what I would do. But that music, it reminded me of what once when I was in Las Vegas and I was at a meeting and I walked through the casino and all of the machines were chiming. And as they were chiming, it was one of the most pleasant sounds I had heard. It was these small bell ringing sounds that were just melody-like and just lovely, but it was all of the machines, and so how could you get them to be in sync like that? And I get what you're saying, that sync and the experience at the moment was just, I stopped actually, and I was like, wow, I wish I had recorded this, because I know they didn't do it deliberately, but this is really fitting, just not what I'm seeing in a casino, but what's in my head right now. And so, but in a video game, I've had the same experience playing something where the music in the background of the video game, and I actually would stop and say, pause while it was happening, to just, because you're right, you drive it and you move it, to say, this is just too perfect. How did you do it?
Yeah.
That's a great example. Because you know what, if you do it on a bigger scale, what this whole thing has been the last 15 years for me has been like that instantaneous synchronization of sound and function and everything that took you in a certain place. And because video gaming puts you emotionally in a state of mind.
Absolutely.
So I'm talking about being in that emotional state of mind just by experiencing what the process was. Wow, guys, I feel good about that now. It makes me feel better.
Good.
It's either that or you accept the fact that just by accident all of this happened, and there was nothing. It's just total serendipity. There was nothing ever at any point, just then an accident after an accident after an accident that ended up being right at the end. Which it could be that too.
Yeah.
I can't prove or disprove either side. But I do sit back and say, wow, how did this group happen, and how did we ever do this project that we just finished? Because I will tell you, to be very humble, we all still do this where we go, did we really do this? Look at a good thing we took a lot of pictures and movies and have good data and recorded every tiny bit of it, because I still have to look back at it and say, we really did that. We bioengineered a lung in a beaker, in a container that we put into the animal, and the animal not only lived, was perfectly fine.
Yeah. Alex, any thoughts on serendipity?
I think there was some, I forget where we were talking about this, Chase, but somebody brought it up. Basically, like, there's so many millions and, billions of little things that happen each day, that the chance that one of them will be some type of crazy coincidence is actually pretty high, even with each one having a probability of, something like minuscule, right?
I think that's what I meant by serendipity is partially a skill of attention. Is you have to pay attention enough to find.
You have to know when you meet somebody that they're important. You have to realize that when you serendipitously do something, it's good. And yes, you do have to attend to it. Although I will admit, at the time, sometimes when I met people, I would have not bet on the fact that immediately that it was part of the research team. Sometimes it took me a little bit more time to make that decision, but then it still came back to, this is the right person. And so we had surgeons that we approached initially that always said to us, when you get a vascular system, come and talk to us. When you get it to the point you prove feasibility, come and talk to us. Until that point, we'd never met a surgeon who said, I don't care that you don't have anything to prove, I believe in it. That to me, that was a little bigger than just serendipity. That was, and I cannot tell you how many surgeons said the opposite to us, who now come forward though now and say, hey, you want us to work on the project? And we're like, no, this crazy surgeon that we work with, who is an amazing cardiothoracic surgeon, who when I watch him, I think there's an art to this work. He's an artist as a surgeon. He is the right person, and he's the one that has to do the next step until we prove without a doubt that we've done what we said we were going to do. And then eventually, the future will be, if we're talking about serendipity, the future will then go to the students who we've trained in the laboratory to do what we call the scientific relay race. To take it from where I take it as far as I can before I retire, and eventually I'm going to have to retire, because nobody can do this forever. But have the students pick up this relay race, and hopefully they have learned how to recognize the serendipity of when it's the right time to do what they need to do and with the right people.
Yeah.
And that's what's next.
Awesome.
Thank you, guys.
Great.
This has been
Well,
fun.
Yeah, Joan, thank you very much. We're so glad to have had you here. If people are interested in following up, seeing what you guys are working on, if they have questions for you, are there, and we'll link everything in the description, but are there public presence places for you or your team that you want to put out there?
And the easy way to find them is to just do a search for Joan, Joan, Nichols, Nichols, at University of Texas Medical Branch in Galveston, UTMB. So if you do Joan and UTMB, all of them will come up. All of the website links, or Joan UTMB and lungs. I know this because that's how people are finding me now. They're saying, oh, I don't have to do much to find everything. So, but I will also send you to add with this some other publications we've done, some other links of things. We also did a CBS Innovation Nation growing lungs segment, and I'll send you that so that you can get it. And
Great.
I did a TEDx talk a couple years ago, growing lungs. And so I'll give you all those links for people to be able to go to and look at this other stuff.
Perfect.
Great.
So those will be in the show notes, everyone. Awesome. Well, again, Joan, thank you very much for your time. And yeah, I'm excited to see what the future of bioengineered lungs looks like.
Great. I am too. But I'll let you know how it progresses, okay?
All right.
Great.
All right, guys. Bye.
Bye. Thanks, Joan.
You're
Thanks.
welcome. Bye-bye.
The Switch is produced by Mojo Filter Media, a company specializing in audio production for video games and interactive media. You can check out our website at www.mojofilter.media.